Good morning again. So this is the final and third part of the metabolism lecture, and this will actually be a much shorter one. This is the easiest of the three. And what we're going to talk about now is anaerobic respiration. Anaerobic respiration is different than cellular respiration because there's no oxygen. As we talked about in the second part of this lecture, anaerobic respiration involves the use of oxygen being the final electron acceptor. Because we're doing anaerobic respiration, the final electron acceptor in the electron transport chain is not oxygen. It does yield less energy than that of aerobic respiration for the main reason that any NADH that you produce or any FADH that you produce cannot go to the electron transport chain. Why? Because the electron transport chain is shut down without oxygen. Now here's the thing. You're going to go through glycolysis. You're going to produce NADH molecules. The cell only has so many available NAD molecules. Because there's only so many available NAD molecules, at some point, if you're going through glycolysis under anaerobic conditions, you're going to fill up all of your NADHs. I'm sorry, all of your NADs to NADHs. When that occurs, there's nothing left. There's no more shuttles. All the shuttles are full. There's no more shuttles to pick up your electrons. When that happens, you can't even go through glycolysis anymore. And if you can't even go through glycolysis, metabolism comes to a complete halt. And when metabolism comes to a complete halt, the cell dies. Okay? Now, unfortunately, that's a scenario that happens in our cells. We are not capable of surviving on anaerobic respiration. But there are many bacteria that can. And what will happen is when they go through glycolysis, they will produce, the 2 ATPs net by substrate level phosphorylation. They will produce those 2 NADHs, and they will make their 2 pyruvic acids. So the pathway is the same that we talked about with cellular respiration. But the thing is that if you have a fermentation pathway, what then will happen is the NADHs will release their electrons back into pyruvic acid. There's an enzyme that will do that. And when they release those electrons back into pyruvic acid, what happens is you end up making an acid or an alcohol, depending on the enzyme that is present. So what's going to happen again with anaerobic respiration is you're going to take your pyruvic acid, you're going to take your NADHs. The NADHs will be able to release the electrons, because there's an enzyme to do that, into pyruvic acid, forming an acid or an alcohol. When that occurs, you have now freed up the shuttle. And the shuttle can then go back and pick up more electrons on glycolysis and then shuttle them back and forth. And because you're able to continue glycolysis, because you're freeing up those shuttles, the cell stays alive. Now, the problem with anaerobic respiration is because you're not involving oxidative phosphorylation, which is how you make a lot of ATPs, you're not going to make as many ATPs under anaerobic respiration. But the cell remains alive. So if we look, this again is going over glycolysis. This is cellular respiration, okay? And what's going to happen with anaerobic respiration, which is what we call fermentation in bacteria. Here's some very key points I mentioned just a few minutes ago. You're going to go through glycolysis, okay? Now remember, you're going to make a net of 2 ATPs by substrate level phosphorylation. We went through glycolysis in the prior lecture. Remember, you actually make 2 and 2, but because you use 2, you end up with a net of 2. You're going to make 2 NADHs, okay? and 2 pyruvic acid molecules. So the pathway is the same, nothing's changed. Now what's going to happen under fermentation is you're going to take these 2 NADHs, as you can see here. They're going to release their electrons back into pyruvic acid, forming NAD, which then can go back and be reused by glycolysis. So it's going to shuttle back and forth between the two. The pyruvic acid will then be converted to some type of fermentation end product. The fermentation end product will either be an acid or an alcohol. Okay. Now, there are different types of fermentation, and the reason why there are different types of fermentation depends on what enzyme is present in the cell to essentially break down pyruvic acid, and take those electrons. So you can either make acids, you can make alcohol, and what I want to do is just kind of show you this little diagram. So, we went through glycolysis, we have pyruvic acid, and you can see we can make different acid products, lactic acid, propionic acid, butyric acid, and then we can make different types of alcohol such as ethanol, butanol. There's many different types of acids and alcohols that can be produced, and this is by no means a complete list. And if you notice, each bacteria is actually producing a different end product. Again, the reason why is that they all have enzymes for fermentation, but the enzymes that they have are different. They're going to make different products. Okay, and this shows diversity amongst the organisms. They're not all making the same thing. And because they all don't make the same thing, we are actually able to use that for testing purposes. We can see what products actually get made by fermentation by the bacteria. And by looking at what products get made, that can help us identify the bacteria, because we know certain bacteria can only make certain products. And in one of the labs that you'll be doing, virtual labs that you'll be doing, goes through these fermentation assays for identification purposes. Now, here's just a list of some of the products that we can make through fermentation. You can make ethanol, acetic acid, lactic acid, citric acid, methane, sorbose, acetone. And if you don't know what a lot of these things are, here's sort of what they are and what their commercial use is. You probably use a lot of these things in your daily life here. Okay. So again, to sort of wrap up metabolism here, with carbohydrate metabolism, you have 2 main options. Okay. You have the respiration routes, where you can essentially go through glycolysis, TCA cycle, electron transport chain, and you can do this because you have the presence of oxygen. And then you have fermentation, where you can go through glycolysis and then a fermentative pathway, and you would do this because there is no oxygen available. Okay. The rest of the slides kind of go over lipid and protein catabolism, meaning breaking down lipids and proteins. What I want you guys to understand is that proteins can be broken down for energy, okay? Because proteins can be broken down for energy, that is something that can be used as a nutritional source. The other thing that can be broken down is lipids and fats. Lipids and fats can be broken down for energy as well. And what happens is when you break down lipids, lipids are typically stored as triglycerides, where you have a glycerol molecule and 3 fatty acid chains. Tri, the 3 fatty acid chains, and a glycerol, triglycerides. When you break the triglyceride into a glycerol and fatty acid chains, what happens is the glycerol molecule will go into glycolysis, and you will continue down glycolysis pathway, Krebs cycle, electron transport. When you break down the fatty acids will get broken down by a process called beta oxidation. And what happens is fatty acid chains are many carbons long. They could be anywhere from 16 to 28 carbons long. And what happens is an enzyme involved in beta oxidation, and this is a much more complicated process, but we're not getting into that. The enzymes involved in beta oxidation will break off 2 carbons off the chain. That 2-carbon molecule that gets broken off is acetyl-CoA. Now, acetyl-CoA should look familiar to you. Acetyl-CoA is the molecule that feeds into the Krebs cycle, okay? So if you have a fatty acid chain that's, say, 28 carbons long, and you break off 2 carbons at a time, that means you can end up making 14 2-carbon molecules out of that 28-carbon chain. In essence, making 14 acetyl-CoAs by beta oxidation. And all of those acetyl-CoAs will then go through the Krebs cycle. Now, if you remember, glucose, you only got 2 acetyl-CoAs. Here, on one fatty acid chain from a triglyceride, remember there's 3, and a glycerol, you're actually making 14 acetyl-CoAs. Then you have the second fatty acid chain, and the third fatty acid chain, and the glycerol molecule. And you can end up making, you know, anywhere from 40 to 60 acetyl-CoAs, all of which are going to get processed by the TCA cycle by breaking down a triglyceride. So instead of making 38 ATPs, you're actually going to make hundreds of ATPs. And that's why they say fat has a lot of energy storage, because there's a lot of energy stored up in fats. Now, what do I want you to know for the test? You don't need to know the pathways for beta oxidation or lipid breakdown. What you should know is, what is beta oxidation? Okay, I would suspect I would see a question on beta oxidation. So you should know that beta oxidation is breaking fatty acid chains down to 2 acetyl-CoA molecules. So you should know that pathway, okay? You should also know that proteins can be broken down, okay? And that's why I'm going to stop at this slide right here, because this really shows you metabolism in a very pared down schematic. But, you know, we primarily study what happens with glucose, okay? And that's really what's in the center here. But glucose is not your only nutrient. Proteins can be broken down into amino acids. Those amino acids then can be broken down for energy. And they can enter, depending on the amino acid, multiple points along the pathway. The lipids can be broken down into glycerol, which will go into glycolysis, and then fatty acid chains, which will go into the Krebs cycle, okay? So out of our 3 major macromolecules here, carbohydrates, lipids, and proteins, they can all yield energy for the cell, okay? And that's where I'm going to stop for metabolism. The remaining slides sort of get into some testing for biochemical tests. You're actually going to have a lab on that. That's lab number 7, which will be in a week or 2, okay? But for lecture purposes, you can stop at this slide, all right? Hope you enjoyed metabolism. It's a wonderful topic. I actually love it. I know most students hate it, but please, please, please do not just read over this once and think you've got it, because you don't. You need to spend some time with this. You will definitely see some open-ended questions. I am looking for details on those open-ended questions, especially on the exam. So make sure you are prepared for those. Okay? Have a good day, guys.